of the vaccine. Now, one of the unique qualities of the Pfizer vaccine is that it has to be kept extremely cold, around minus 94 degrees. Here now to discuss the challenges associated with the cold storage of the Pfizer vaccine is Salk Institute for Biological Studies professor Susan Keck. Susan, good to talk with you tonight.
You too. Good evening.
You know, ever since I've been reading about this data is certainly exciting, but I'm interested to find out more about why does it need to be kept so cold?
Yeah, so this vaccine, as well as the Moderna vaccine that just released its very high level of protective efficacy as well, are mRNA vaccines, which you've been hearing about. And the mRNA vaccines are actually fairly simple in their design. They're basically just two components. You have the mRNA molecule, which is why we call them mRNA vaccines, but you also have a lipid capsule that is as a part of the vaccine. And so the mRNA molecule is what is used, or cells use, to synthesize proteins. And so the mRNA is a template, it's a coded molecule that our cells can then read and start to generate proteins. And in this case, the vaccine is expressing, it's producing the protein for the SARS-CoV-2 spike protein. And this is really important because the spike protein is what the virus uses to allow entry into our cells. And so the vaccine is trying to help our bodies produce this virus spike protein so that then we can generate an immune response to it to hopefully neutralize and prevent the entry of the virus into our cells.
But our bodies are, you know, close to 100 degrees warmer than what this vaccine will be kept at. So how does that work together?
Right. So the vaccine, the reason why they need to be kept so cold is because the mRNA molecule is actually quite unstable and it can be easily degraded. And that's why it has this lipid capsule that the mRNA molecules lives in. It lives inside this capsule. And this lipid capsule is basically kind of like a fatty, waxy capsule. But that is also really important for allowing this vaccine to be delivered. The lipid capsule helps the vaccine be delivered into our cells so that we can produce the protein. But the lipid capsule itself is actually also quite unstable at higher temperatures. So it's actually the cold temperatures, while the mRNA molecule itself survives better in cold temperatures. The ultra cold temperatures are actually needed for the stability of this lipid capsule that houses the mRNA molecule.
Okay, so if it's, you know, let's call it fragile, because I'm guessing then if this vaccine gets warmer, that means its efficacy is going to go down, or maybe it has no effectiveness. So what does this mean for distribution? Because to have a fridge that's able to keep something that cold requires a lot of red tape.
Yeah, so there are freezers that we have that can reach these temperatures that are used often in industry and in medicine and in research. But you're right, they're not the most common types of freezers that you would have. It's not like the freezer that you have in your house that can reach these ultra cold temperatures. But there are freezers that we can use for transport. So the problem's not unsurmountable. It certainly makes it challenging, but we do have the ability to transport the vaccine at these cold temperatures. I think what's going to be another issue is then, you know, what happens if the vaccines, if they're not, how long they're able to be kept, if they're not able to be kept at these temperatures. Like you said, they're quite fragile. So there's not going to be much time if the vaccines are exposed to higher temperatures for them to last.
And I'm curious, would you be able to tell immediately if a vaccine has gotten too warm and it's no longer good?
Yeah, I don't know if there is any inside internal controls that they have for the vaccines at this stage to know if they've been raised to a temperature. You could, I imagine, do that with the packaging that the vaccines are housed in. That could have some kind of a temperature indicator if the product in the vials had been exposed to a higher temperature for too long.
Yeah, very interesting. Class has certainly been in session today with Professor Keck. I really appreciate your insight tonight.
Thank you.